Welcome everyone, and thank you for attending this webinar entitled Is RNA Medicine the Next Paradigm Shift? My name is Anne Rodalek and I will be sharing today this webinar. I'm organizing webinars on behalf of the Controlled Release Society Benelux and France local chapter, a non-profit organization that aims to exchange knowledge about between scientific and industry experts, sharing both of their expertise on the same topic. So I would like to thank Precision NanoSystems and especially Jane Aleva and AJ Joni for collaborating with us in organization of this webinar and for hosting us on their platform today. The webinar will consist of 3 parts. First, we will have Samuel Clarke from industry side, followed by a talk by Peter Vader from academia, and finally we have a Q&A session. With that, I hope you will enjoy the next hour, and it's my pleasure to introduce you to Samuel Clarke, the R&D Director of Precision NanoSystems, that will discuss with us his recent work on the development of a mRNA nanoparticle from discovery to scale-up production. Samuel, the floor is yours.
Thank you for the introduction, and it's exciting to have the opportunity to speak about some of our work in this area. Okay, so let's get started here. The title for my talk, as Anne mentioned, is around robust and scalable manufacturing of a model mRNA lipid nanoparticle therapeutic. And in terms of the talk here, the outline is around, I'd like to start with the brief description of the nanoparticle therapeutics landscape, a little bit of background, and then I would like to focus on discussing a couple of technologies that are really important for the development of nanoparticles. One is lipid nanoparticle formulations for nucleic acid delivery, and the second area is microfluidics for scalable manufacture of these lipid nanoparticles. So I'll go through the concepts and some of the background there. And then finally, at the end of the talk, what I'd like to do is go through a data set showing how you can use these technologies to rapidly develop RNA-based drugs. So at Precision NanoSystems, our vision is to accelerate the creation of transformative medicine that significantly impacts human well-being. And we, as a company, we work in this field of nanoparticle therapeutics, really recognized as an industry leader and a gold standard in the market. And so nanoparticle therapeutics are really poised to make a huge impact in the area of medicine. They're being applied in the area of vaccines, which is of course very timely now with the COVID pandemic. They're being applied in the area of cell therapy, in immuno-oncology, for small molecule delivery and targeted therapeutics, and for RNA and DNA therapeutics, as well as CRISPR gene editing. So really there's a wide breadth of application for nanoparticle therapeutics. And then as a company and with our products, Precision really is involved with many different groups in these areas. We have more than 400 of our systems deployed worldwide, work with more than 90 academic groups, more than 125 industry groups. So really we're working at the forefront of this field. And so for the talk today, I would like to focus on the application area of RNA medicine, which really a tremendously exciting area where RNA could lead to a huge paradigm shift in terms of how we treat disease. Some recent examples in the RNA medicine area include Onpattro, which is a clinically improved drug. This is based on single interfering RNA encapsulated in lipid nanoparticles and used to treat a liver disease. And this works by suppressing the expression of a protein in the liver. Another example is in the area of gene editing, where companies like Intellia Therapeutics, they're using lipid nanoparticles to encapsulate the CRISPR gene editing machinery in the form of messenger RNA and guide RNA to permanently edit out genes associated with disease. And then the third example would be the area of RNA and genetic vaccines. So for example, Precision NanoSystems have recently announced a partnership with CanSino Bio, where we're working together to develop a vaccine for COVID-19 based on the principles of self-amplifying RNA and RNA-based approaches for lipid nanoparticles. And so overall, RNA is revolutionizing rare disease, infectious disease, and cancer therapy, and is a really exciting field. So Precision NanoSystems, we have really two core technologies. One is a lipid nanoparticle technology. I'll speak about our GenBoy ILM reagent technology in this talk and show some data in this area. And the second is a manufacturing technology based on microfluidics and our nanoassembler platform. So this is for the manufacturing of nanoparticle-based therapeutics. So combined, the lipid nanoparticle technology and the manufacturing technology really provide a full solution for the development of this class of therapeutics. So I'll start by introducing lipid nanoparticles and provide more background here, and in particular for their use for nucleic acid delivery. This slide provides an overview of and a description of lipid nanoparticles for this application. For those not familiar, lipid nanoparticles are designed from chemically defined components. There's typically 4 to 5 components. They're designed to protect a nucleic acid payload from degradation, and they have very low toxicity. They are designed for effective delivery to primary cells, both in vitro as well as in vivo. In the case of in vivo, when administered systemically, they typically get sequestered to the liver, and so there have been lots of applications, the use of these nanoparticles to treat liver disease. The nanoparticles are designed to facilitate cellular uptake through a receptor-mediated process, and they're designed to degrade in a pH-sensitive way. So as they're endocytosed and enter the endosomes, the change in pH disrupts the nanoparticle and delivers the nucleic acid to the cytoplasm. And so this technology has been demonstrated now for a wide range of nucleic acids, including plasmids, mRNA, and both small and large RNAs. So GenBoy ILM is a product from Precision NanoSystems that's a lipid nanoparticle formulation. And it is designed specifically for RNA applications. This includes for siRNA, for sustained gene knockdown. The example on the left here shows the use of GenBoy ILM to knock down factor 7 in a mouse model. So we can see here, compared to controls at 100% level of the gene, samples that contain nanoparticles with siRNA for factor 7 led to an almost complete reduction in the expression of this gene in mice. And then a second example here on the right-hand side is with messenger RNA for gene expression. So this example shows that nanoparticles encapsulating a messenger RNA encoding for luciferase are sequestered to the liver in mice, and then lead to high expression of the luciferase reporter gene. Overall, this data shows a couple of examples of how GenBoy ILM and how lipid nanoparticles can be applied for these RNA applications. And so then I wanted to move on to talk now about the manufacturing side of these nanoparticles and provide a little bit more background here. And so I think what's important for everybody to understand is that nanoparticles are more complex than previous generations of drugs. And this table here shows a comparison of a nanoparticle, of a lipid nanoparticle encapsulating RNA with drugs in the form of antibodies, as well as small molecules. And the point to make here is that these RNA lipid nanoparticles, they're both quite a bit larger than traditional drugs, as well. They are more complex and have more different types of molecules. And so what becomes very important is the manufacturing process for these nanoparticles. In the case of these RNA lipid nanoparticles, the process to assemble these is a self-assembly process. And one of the key aspects is really careful control over the mixing steps involved in forming the nanoparticles. And this schematic here shows an example of the self-assembly of an RNA molecule with the lipid components of a lipid nanoparticle over time. And so really a key step behind this is that mixing step that has to be carefully controlled. And so one of the key technologies, the core technologies that we've developed at Precision NanoSystems are microfluidic-based approaches to control this mixing step and to provide a very controlled self-assembly for lipid nanoparticles. This schematic here highlights the concept where the manufacturing process involves combining the lipid components of the nanoparticle with the nucleic acid payload in a microfluidic device, carefully controlling the flow of those two reagent streams using microfluidic design, and then rapidly mixing again in a microfluidic design to enable millisecond mixing and this very high degree of control over the nanoparticle formulation and formation process. So we have shown really the advantages of microfluidics for the production of these type of nanoparticles in terms of ease of use, as well as reproducibility, and further the ability to control the size and physical chemical properties of the nanoparticle to a very high degree. This approach using microfluidics to manufacture lipid nanoparticles has evolved from other methods to produce nanoparticles. This slide really just describes a bit of the evolution of the approaches for manufacturing nanoparticles for therapeutic applications. Earlier techniques were based on high energy techniques such as sonication or extrusion. These techniques were complicated, they had limited applications, challenges with reproducibility and scalability. Macro mixing techniques emerged from there, and these did improve aspects such as scalability, but still had limited applications and challenges with reproducibility. And over the last 10 years or more, microfluidics approaches have really emerged as an effective approach for producing nanoparticles with improved reproducibility and really expanded range of applications. And so now today with the next-gen microfluidics technology that we have at Precision NanoSystems, we've really designed this to overcome all the key challenges associated with producing nanoparticles and doing this in a reproducible and scalable way. In terms of the next-gen microfluidic technology at Precision NanoSystems, we have this implemented across a range of instruments. The Spark, the Ignite, the next-gen Blaze, as well as our GMP system that allows for the production of nanoparticles at a wide range of different scales and supports the continuum of the development of drugs from discovery and development all the way to clinical development. Okay, and so in the last section here, I would like to highlight a data set showing the use of both the GenBoy ILM, lipid nanoparticle technology, as well as the next-gen microfluidics technology. And so the approach we took here was to develop a model messenger RNA therapeutic. we selected one based on the molecule erythropoietin, or EPO. And this is a useful therapeutic for the treatment of a disease called, or a condition called anemia. Okay, and so this slide describes overall this concept. The traditional approach right now to treat anemia is with a recombinant protein, recombinant EPO protein, which can be administered as a drug. So to demonstrate the RNA technology, what we did is we took a erythropoietin encoded mRNA and packaged that into a GenBoy ILM lipid nanoparticle using the next-gen technology for production. And so this EPO lipid nanoparticle can therefore have a therapeutic application when it's administered into the blood, and then it's delivered to the kidney, where it can produce the EPO protein and lead to a correction in the anemia. And so I'll show some data in vivo data showing how this therapeutic can work. So for the data and to describe the approach that we've taken to generate these nanoparticles, I just wanted to highlight the nanomedicine manufacturing process. The approach we take at small scale is to model a process that's applicable at the GMP scale. And so what it allows us to do is to rapidly transition from the discovery phase into preclinical development, and then into clinical development in a very fast way. So this schematic shows the main unit operations associated with producing these nanoparticles. They include the microfluidic mixing step that I've described, dilution, downstream processing using tangential flow filtration, sterile filtration, and final fill finish to create the drug product. So the data that I'm going to show you was generated using this type of process. And specifically in terms of the experimental design for the study here, what we have done is we've produced samples of the mRNA lipid nanoparticle encoding for erythropoietin on the Ignite at small scale, at about 4 milliliters, on the Blaze system at larger scale, up to 55 milliliters, and then on the next-gen GMP system, again at a larger scale, to demonstrate the rapid development and scalable manufacturing of this type of nanoparticle. And so this data here shows a small scale characterization of nanoparticles produced at small scale on the Ignite system, and compared to a clinical benchmark formulation incorporating the MC3 ionizable lipid, which is a formulation that's often benchmarked in industry. So this data here on the left-hand side shows the relative pKa of these nanoparticles, and using an assay called TNS. So it shows a pKa of about 6, and overall comparable results between the GenBoy ILM lipid nanoparticle and this clinical benchmark MC3. When we look at the size of these nanoparticles, this data shows comparable size across the MC3 benchmark formulation, as well as the GenBoy ILM formulation, at 2 different N/P ratios, which are the ratio of the mRNA to the lipid. And then we can look at encapsulation efficiency of these nanoparticles, their ability to encapsulate the mRNA. Both cases across the benchmark and the GenBoy ILM sample, very high encapsulation efficiency above 95%. So this really highlights how GenBoy ILM nanoparticles encapsulating EPO RNA have good physicochemical properties. And then we can take this to an in vivo study to look at biological activity and a therapeutic effect. And so when injected into mice, we looked for serum protein levels of erythropoietin 6 hours post-injection, able here to demonstrate that both the GenBoy lipid nanoparticles and the MC3 benchmark formulation led to similar levels of EPO production in mice. And then we looked further for a therapeutic effect, looking at erythrocyte levels 7 days post-treatment, found that both the GenBoy ILM lipid nanoparticles and MC3 benchmark lipid nanoparticles led to an increase in RBC production, which is the expected effect of from the production of erythropoietin in vivo. And then as a final data set, I wanted to highlight the ability now to take this mRNA lipid nanoparticle formulation at small scale and rapidly scale that up using the next-gen manufacturing technology. So this data on the screen here shows size on the left-hand side, and then a comparison of samples as a function of the scale. And so when we produce samples on the Ignite at small scale, at larger scale on Blaze, and then at large scale on GMP, found that the size of the nanoparticles are very similar, around 60 nanometers, as well the polydispersity very low, less than 0.1. Across scales, the encapsulation efficiency is equivalent. And across scales, when we look at composition using HPLC analysis, we can see that the composition of the nanoparticles is consistent. So from this data, we can conclude that these EPO-encoded mRNA using GenBoy ILM, they all have similar size, polydispersity, and other physicochemical characteristics across all scales. And finally, we looked at the, again, the in vivo biological activity of these lipid nanoparticles, comparing performance across scales. On the left-hand side here, again, this is an assessment of EPO production in serum. And we can see that the levels of EPO production are very similar across all the scales of nanoparticles produced. And further, when we compare downstream processing methods using ultracentrifugation as well as tangential flow filtration, levels of EPO production were similar as well. And then again, when we looked to for an increase in production of red blood cells 7 days post-treatment, we saw similar increases across all nanoparticles produced at different scales, as well as with different downstream processing methods. So what this biological data demonstrates is that, again, the properties and the performance of the nanoparticles are very similar or equivalent across all scales of production. So to conclude, what I wanted to highlight here with this case study is that we've been able to generate therapeutic levels of EPO in mice using these erythropoietin encoded mRNA lipid nanoparticles. The GenBoy ILM formulation lipid nanoparticle mix provides comparable performance to a clinical benchmark shown in our benchmarking studies, and further can be used to produce high quality lipid nanoparticles for proof of concept of preclinical studies with any therapeutic mRNA. And then finally, the next gen technology for manufacturing, it enables rapid production and seamless scale up of nucleic acid lipid nanoparticles. And so these technologies really provide a way to accelerate drug development all the way from bench scale towards clinical scale GMP manufacturing. Finally, I just wanted to quickly acknowledge P&I's R&D science and engineering teams. This is the team that has helped to develop these products and generate the data that I've walked through today, as well as P&I's departments and our many collaborators and scientific advisors who helped make this possible. So with that, I would like to introduce Dr. Peter Vader, who will talk to us about his work in the area of extracellular vesicles for RNA delivery. So I'm looking forward to hearing an update from Peter. Thank you.
Yeah, thank you very much, Samuel, and thanks for hosting to Precision NanoSystems and to the CRS Benelux France chapter for the kind invitation to present some of our work. There we go. So my lab is interested in the use of extracellular vesicles for therapeutic applications, and today I would like to share some of our recent work on therapeutic RNA delivery. So, as been introduced by the previous speaker already a little bit, is that RNA-based therapeutics have unprecedented potential for the treatment of many diseases. And when we talk about RNA-based therapeutics, these include, for example, the mediators of RNA interference, siRNAs and microRNAs, mRNAs, as well as the RNA components of the CRISPRCas9 machinery. And as a class, we recognize that they allow us to silence, express, or edit virtually any gene in our body. And this, of course, gives RNA therapeutics a enormous therapeutic potential. But we also recognize that RNA molecules are large molecules, negatively charged, very fragile. So they typically require delivery to the site of action, which is the cytosol of the target cells. And we know this is rather difficult. And over the course of the last few decades, delivery systems have been developed that aid in delivering these RNA therapeutics into disease cells. And this has been exciting times because we've witnessed the approval of Onpetro. But we also know that for most of these lipid nanoparticles and other carriers of RNA therapeutics, their efficiency is really limited to delivering to the liver and perhaps spleen and lung to a lesser extent. So there is still a need for improved systems, especially for targeting other organs. And for this reason, we and many others have started to look at endogenous membrane-enclosed particles called extracellular vesicles. So these vesicles are a heterogeneous population of particles that are secreted by various cell types. They are between 30 to 1,000 nanometers in size, peaking around 100 nanometers. And besides having a lipid bilayer, they carry proteins and RNA derived from the cells that secrete them. And since 2007, we've learned that actually these biomolecules that are carried by these extracellular vesicles can be transferred from one cell to another and processed by the recipient cell, leading to phenotypical changes. So these vesicles seem to be doing what we are aiming for, and that is deliver their cargo into recipient cells in a functional manner. But there's other potential advantages that these extracellular vesicles could have as compared to synthetic systems. For example, they bear no or low immunogenicity, especially, of course, when harvested from a patient's own cells. They're intrinsically stable in the circulation, and they might utilize native mechanisms for targeting, uptake, and trafficking inside the cell, although this really has to be shown still. And then lastly, when taken from the appropriate cell types and when used for the appropriate application, these vesicles, through their endogenous cargo, may already have intrinsic therapeutic properties. For example, we've shown that if you inject vesicles derived from cardiac progenitor cells into the myocardium right after an infarction, you can decrease an infarct size in mice in this case significantly. So the vesicles have intrinsic protective properties. So for this reason, we got interested in the use of these vesicles for RNA delivery. And of course, when you want to work with these vesicles, you first have to get your hands on them. As I mentioned, these are secreted from cells and also from cells in cell culture. So what one can do is take the conditioned medium from cells, take that, do a few low-speed spins to pellet the cells, and take conditioned medium and isolate the vesicles using size exclusion chromatography, in which you separate the vesicles from the non-secure secreted material. And what we typically then do is characterize these vesicles. We do Western blotting to look for specific marker proteins that we need to get enriched in our vesicles as compared to a cell lysate, and other marker proteins, for example, connexin, which is a ER marker, we do not find in our vesicles, but we do find it in the cell lysate. So we do this as a sort of quality control measurements. we find that these vesicles are typically 100 nanometers in size, as I mentioned, and under the electron microscope, they have this typical encapsulated morphology, and the size matches what we find with MTA. So then, of course, what you want to do is load these vesicles with our RNA of choice, a therapeutic RNA. And the first thing we tried is to see whether we can use the endogenous machinery of the cell. So we know that these vesicles naturally carry RNA molecules. So we thought, well, hey, if we express something in the cell that secretes these vesicles, will the RNA then also end up in our vesicles? So we did a very simple experiment. In this case, we tried to load siRNAs into these vesicles. So we transduced donor cells, X2-free T cells in this case, with an expression factor that would allow us to express short hairpin RNA into the cytosol of these cells. And then we were hoping that the cell would spontaneously package these RNA molecules into a variety of different types of extracellular vesicles. And indeed, we could find them back, but at very, very low efficiency. So typically, we could only find one siRNA molecule per 10 or 100,000 EVs. And this is, of course, way too low to have any significant effect in reporter cells, especially when we think in terms of therapeutic benefits. So then we moved on to a slightly different approach to be able to show that vesicles can in fact deliver RNA to recipient cells. So we hypothesized that if we would take a hydrophobically modified siRNA, in this case a siRNA tagged with a cholesterol molecule, that by simple mixing, the cholesterol would anchor into the membrane of the extracellular vesicles, and in this way, if we would add these vesicles to recipient cells, the vesicles would be taken up and the siRNA would be dragged inside with it. Of course, the siRNA would be facing the surface of the vesicle. That's why we used a quite heavily modified RNA molecule. But this could allow us to show proof of principle. So we did that experiment, and we first optimized the loading conditions for optimal loading of siRNA into these vesicles or onto these vesicles, really. So we looked at incubation temperature, incubation time, the volume, and the ratio between the vesicle and the cholesterol conjugated siRNAs. And then we found out that the optimal conditions, I'm not going to spend too much time on this slide, but were 37 degrees, incubation for 1 hour, at a specific volume, and a specific ratio between the two. We could load more. This was the optimal that we found was 11 siRNA molecules per EV. We could load more, but we also found that in a way obstructed the delivery efficiency. So this ratio seems to be optimal to have maximal delivery efficiency. So we then moved on to see whether indeed this could give us knockdown in the recipient cells that would take up these vesicles and process the siRNA. So we did an experiment where we took these vesicles, we loaded them with HUR siRNA. HUR is a potential anti-cancer target. And we added these vesicles loaded with different amounts of siRNA onto recipient cells. And here we could clearly see a dose-dependent effect on HUR expression. And that effect was dependent on both the siRNA sequence, because control siRNA loaded onto vesicles had no effect, as well as the presence of the vesicles themselves, because a gymnosis experiment in which we just added the siRNA to the cells did not show any effect. So indeed, we found dose-dependent knockdown of our target when the siRNA was loaded onto the vesicles, and this effect lasted for up to 6 days. So clearly this provided proof of principle for us that these vesicles could carry RNAs into recipient cells, but not very efficiently. These concentrations are quite high in general. But still, there was another lab that around the same time published this paper that showed actually with a similar approach, they could silence Huntington mRNA in the brain after a local injection. So perhaps there is a future for this approach after all. But we continued our studies because we were really interested to see how efficient actually these vesicles could deliver such RNA molecules. And we thought, well, we can use the approach that I just showed you, having the siRNA on the outside. Of course, it's not really fair because vesicles naturally carry the RNA in their lumen. So if you really want to study how efficient these vesicles deliver their cargo and how this really works, we would have to deal with a situation where we have very, very low RNA loading efficiency. This is what we typically find for different RNA subtypes. So instead of improving the loading, we thought, hey, maybe we can design a reporter system that would allow us to study this vesicle trafficking at a very, very sensitive manner. So for this, we turn to CRISPRCas9 machinery. We are all aware of this. So we know that the Cas9 protein can be guided by a guide RNA to recognize a specific sequence in the DNA, in genomic DNA. And when this happens at a specific place in the DNA, this Cas9 protein will make a double-stranded break. And we also know that the cell will try to repair this break, but it's error-prone. The cell will try to use non-homologous end joining, which leads to errors, often leads to deletions or small insertions in the DNA and thereby to frame shields. So we try to make use of this trick in our reporter system by having a construct in which we expressed mCherry, but also a linker sequence which contained a Cas9 target with a specific targeting guide RNA. So the idea was that if Cas9 was present in a cell, as well as that specific guide RNA, that the DNA in the linker region would be cut, and then we had two different types of GFP out of frame. But in the case the DNA would be repaired and frame shift would occur, either one or two nucleotides out of frame, we had GFP being expressed. So basically, if guide RNA was present, Cas9 was present, we would see GFP coming into frame, and we would see that in our recipient cells. And to validate that, we transduced HEK cells in this case with a Cas9 protein as well as the targeting guide RNA. And well, in a normal situation, we only see the mCherry. Of the transduction of both the protein as well as the targeting guide RNA, we observed efficient GFP expression on these recipient cells. So that was our reporter concept, and how do we envision then using this for studying vesicle-mediated transfer. So here we have our donor cell. This is the cell that expresses the guide RNA and secretes the vesicles. And the idea is that some of these guide RNA molecules that are expressed in the donor cell would end up in the vesicles. And indeed, we could confirm that by RT-qPCR. And then if we had recipient cells, so reporter cells, which express the Cas9, as well as the stoplight reporter expression. With the idea that when these cells would take up these vesicles and process the guide RNA in a functional manner, that these cells would at some point start to express GFP. So with this cell, with this reporter, we could single out the cells that had taken up vesicles containing guide RNA in a single cell accuracy. So of course, we had to show that this worked. So we did an experiment where we co-cultured two different cells, donor cells, MDA MB231 cells containing the guide RNA, and stoplight reporter cell HEK293T cells expressing also the Cas9. And when we cultured these cells, but we let these cells, the MDA cells, express a non-targeting guide RNA, nothing happens. But when we express the targeting guide RNA, we could see that the targeting guide RNA was transferred from the donor cells into the reporter cells, and we could see that these cells start expressing GFP. Here's a quantification of that. And there was dose dependency in this effect, because the higher the ratio between the donor and the reporter, the more efficient this transfer became. So not all cells turn green, of course, but that's again because this efficiency of loading is still very, very, very low. But on the good side, we can study this now on a single cell level. So what can we do with this? Well, for example, study whether vesicles communicate between every single cell. So we again took our MDA donor cells and then co-cultured them with various recipient cells. You see a bunch reported here. And then try to see whether indeed all these cells would take up these vesicles and process the guide RNA in a functional manner. And in fact, they did, although there were quite striking differences in terms of efficiency. For example, P47D reporter cells have quite efficiently taken up vesicles, while other, for example, HeLa cells did that to a much far less efficient extent. So there were differences, but all reporter cells that we tested in the end took up these vesicles in a functional manner. So what about the donor cells? Now we varied the donor cells. So again, we took HEC cells as reporter cells, but then used HMAC1, H-short MSCs, or HEC293T as donor cells. And now, strikingly, there were differences here. So for HMAC1 and H-short MSC donor cells, we saw again that these cells communicate with each other. When we used HEC donor cells and HEC reporter cells, we saw no green cells appearing. So this suggests that indeed HEC cells do not talk to other HEC cells in this experiment. So that tells us something about the targeting efficiency of these vesicles, or that these HEC cells simply do not carry the guide RNA. So that's something we still need to follow up on. But at least it shows what we can do with the system. So this was all in co-culture. It's basically mimicking the physiological situation where two cells are grown together, talking to each other. But of course, from a drug delivery perspective, the way to study this is using isolated EVs that you would add to recipient cells after isolation. So therefore, again, we isolated EVs from cells expressing the guide RNA using the approach that I previously showed. And again, we characterize these vesicles. So again, similar size, this express the proteins that we want and not the ones that we don't want. If we label them with a fluorescent label, we see that our reporter cells internalize them quite efficiently. But what about the guide RNA? Was it also transferred to these recipient cells? And the answer is yes, of course. When we took vesicles carrying a non-targeting guide RNA, nothing happens. But when we took vesicles carrying a targeting guide RNA, we saw these green cells appearing over time, showing that these cells have functionally taken up these vesicles and processed the guide RNA so that we could find these the reporter activity. And this effect was dose dependent. So what else can we now do with this reporter system? Well, first thing we did is really try to find out how this vesicle-mediated RNA transfer works. And we only started doing this very recently, but you can envision studies like this, where we, for example, transfect siRNAs into our donor cells or our reporter cells before we do the co-culture to look at which genes are actually involved, either on the donor side or on the reporter side, in transfer of RNA. And as an example, here we knock down various genes in the reporter cells that are known to be involved in endocytosis or intercellular trafficking of endosomes, because we think that these vesicles are taken up through endocytosis and then at some point fuse to release their guide RNA into the cytosol. And we could show that, for example, caveolin, a mediator of caveolin-mediated endocytosis, or PEC1 or REC1, these are the key components of the endocytic machinery. If you knock those proteins down, then this RNA transfer via EVs is drastically reduced, suggesting that indeed these types of proteins have a role in this. And this is something we're studying now in more detail. So with that, I would like to come to the conclusions. And I hope that I've convinced you that vesicles can actually functionally deliver RNA to target cells. It's something they naturally do, although that's still under debate. But in our hands, we see that they can do it. The physiological relevance of all that is still highly debated, but they seem to be able to do it. We designed a reporter system that allows us to study this in a bit more detail. And that, for example, has learned us that under certain circumstances, these vesicles seem to outperform LNPs in terms of delivery efficiency. And I hope to show you a lot more on that in the future. So where does it leave us? I think it shows us two things. So loading was terrible for these vesicles. So if you want to use these vesicles themselves for therapeutic studies, then we need to improve the loading of these vesicles with therapeutic RNA drastically. But on the other hand, if EVs indeed under certain circumstances outperform these LNPs, then it may be good in studying these vesicles and seeing how these deliver their cargo into recipient cells in a bit more detail, perhaps finding out what is the cargo and what are the players on the vesicles that makes that so efficient, and then perhaps try to improve synthetic systems through EV-inspired engineering. I think that's the end of my presentation. I just want to mention that we're involved in 2 different European consortia focusing on RNA therapeutics, one targeting the brain, that's B-Smart, and one recent one on mRNA nanomedicines, where we try to do cancer vaccinations and regenerative medicine in the heart, the Expert project. So if you're interested in finding out what we're doing there, please visit these websites and come in contact with us. I would like to thank the CRS again, the Benelux France local chapter, for the invitation. If you want to know more about what they're doing, please visit their website, follow them on Twitter. And I would like to thank the people who did the work that I've presented today, and our funding bodies for helping out. Thank you very much.
Thank you, Peter, and thank you, Samuel, for those 2 very interesting talks. We will now have to close the Q&A session and the webinar. So thank you both for our 2 speakers for sharing with us their knowledge and their latest results. If you have any question left, don't hesitate to, we'll try to follow up, and don't hesitate to share them with the CRS BNLF social channel. And if you like this webinar, more are coming with Precision NanoSystems. So you can see on the slide, the next one is on June 3rd. It's at tea time every other Wednesday. And don't hesitate to follow the CRS BNFL local chapter to see our future presentation. You can follow us on LinkedIn or Twitter. With that, thank you all, and bye-bye.